Good morning, dear students of class 12 science. Welcome to biology class. We're dealing with the chapter strategies for enhancement in food production. So in the last class, we have learned about the plant breeding for disease resistant. Okay, where we have learned about the conventional breeding technique program. We have learned about the mutation breeding program. And we have gone through some examples where we have learned about the common disease resistant crop varieties which are developed in India. Then we have also gone through the plant breeding for development or developing resistance to insect pests, where we have seen the common pest resistance varieties. Clear? Then we have also learned about the plant breeding for improved food quality. So now in today's class, we'll begin with the topic biofortification. Okay, so what is biofortification? It's in the page number 517. Okay, page number 517. So the topic for the day is biofortification. Then we'll go through the plant tissue culture, then single cell protein. Okay, so first of all, what is biofortification? So go with the definition that breeding of crops with higher level of vitamins and minerals or higher proteins and healthier fat is called biofortifications. Yeah, so whenever we used to go with the breeding of plants or the breeding of crops which will have a huge or higher level, let's say, or better quality with adequate amount of vitamins, okay, or minerals, proteins, or fats, then those crops which are developed by this process is known as biofortifications. So in biofortifications, we are producing the crops with higher level of vitamins, okay, minerals, proteins, and fats. Yeah. Now, this particular process, it does not involve genetic engineering. Yeah. So why are we going for biofortification? What are the main objectives of biofortifications? Clear. So breeding for improved nutritional varieties is carried out with the objectives of improving number one, protein content and quality. So mainly in the crop you used to improve the quality and the quantity of the protein. And number two, used to improve the oil content and the quality, the vitamin content number three, and number four, to improve the micronutrient and the mineral content. So these are the main objective of biofortification. Clear? Now, there are certain hybrid crop varieties with improved food quality which has been developed recently. Okay, so we have got few crops which have been developed recently, right, with the improved quality. Let's see. The first one, let's go with the maize hybrids. All right. So in the maize hybrids, they have got the amino acids in double amount. Clear? Compared to the normal one, normal maize varieties, those maize hybrids we have got the double amount of amino acids, mainly lysine and tryptophan. Wheat variety Atlas 66 has got a higher protein content. Clear? And has been used as a donor for improving the cultivated wheat. Remember the name Atlas 66, the name of the wheat variety. Then we have got an iron fortified rice variety containing over 5 times more iron as in common varieties which has been developed recently. Okay, so the IARI means Indian Agricultural Research Institute. Remember this name, IARI, Indian Agricultural Research Institute in New Delhi has released the following vegetable crops which are rich in vitamins and minerals. So let's see. Vitamin A enriched crops are carrot, spinach, and pumpkin. Vitamin C enriched crops are bitter gourd, bethua, tomato, and mustard. Then calcium and iron enriched are beans and garden peas. Okay, so these are the particular name of the vegetable crops which has been enriched with vitamins and the minerals developed by IARI. Okay, so we have finished about biofortifications. So here you have to remember the definitions, the main objectives, and the examples or the name of the few varieties of the crops. Clear? Okay, next one, plant tissue culture. Okay, so what is plant tissue culture? So plant tissue culture is a technique of growing plant cells, tissues, or organs in a sterile cultured medium under the controlled environmental conditions. Okay, so it is mainly a technique where we used to grow the plant cell, or we used to grow the plant tissue or the organs of the plants in a sterile conditions, free from the germs, under the controlled environmental conditions. Okay, Gottlieb Haberland in 1902 studied the technique of plant tissue culture. So go with the page number 517, you will find the history of plant tissue culture also. Clear? Now, the plant parts is very important. Okay, the plant parts which are used for the plant tissue culture, they are known as the explant. So any part of the plant which are which are being used for the plant tissue culture, they are known as explant. So kindly remember the name or term explant, explant. So what is explant? Any part of the plant which are being used for the plant tissue culture is known as explants, and these explants are grown in a solid, semi-solid, or the liquid medium. Okay, the solid culture medium contains inorganic cells to provide essential macro and micronutrients, organic nutrients such as glucose, sucrose, or fructose to provide energy and amino acids, glycine, and vitamins. Then growth hormones like auxin, gibberellin, cytokinins, which are the plant hormone. Okay, then agar-agar or gelatin to make the medium solids. So these are the particular constituents of the solid culture medium. Clear? Okay, next one. Now, procedures of tissue culture. So we'll go with the procedure and the application of tissue culture. So procedure of tissue culture is in the page number 519. Clear? Now, what is the procedure or the process of plant tissue culture? What are the steps it involves? So first step that's involved in plant tissue culture is selection of explant. Explant means any part of the plant that is used for the plant tissue culture. Stems, okay, any part. Sterilization, second step. Third step, transfer of explant to the culture medium. Fourth one, callus formation. And the fifth one, organogenesis and formation of plantlets. Okay, so first of all, let's see the selection of explant. Now the explants may be any part of the plant. I'm telling you that, right? So it may be the root tip, the shoot tip, the bud, beauty bud, the leaf, okay, the inflorescence, the petal part, the anther, ovule, ovary, or embryo. So any part can act as an explant which can be used for the process of plant tissue culture. So we have to select the explant that needs to be used for the plant tissue culture. Then second one is sterilization. The explants, the explant that is selected, okay, the explants, the culture vessels, the culture media, and the instruments are to be made free from the microbes. That is called sterile medium. Okay, which has to be free from the microbes. And this particular process is called sterilization. Okay, and mainly for sterilizations, we used antimicrobial chemicals such as chlorine, water, okay, or sodium or calcium hypochlorite solution. So what are those chemicals? Antimicrobial chemicals which are being used to kill the microbes. It can be chlorine water or sodium or calcium hypochlorite solution, and this is called surface sterilizations. The vessels, okay, culture medium, and the instruments are sterilized with steam, dry heat, or autoclaving at 120 degrees Celsius for 15 to 20 minutes. And this is called complete sterilization. So we have got two process or two steps under sterilization. First one is surface sterilization, which will be followed by the complete sterilization. In surface sterilizations, what we are doing, mainly the explants, the part of the plant that is being used for the culture, needs to be treated with antimicrobial chemicals, okay, which is called surface sterilization. After that, the vessels and the instruments which are being used needs to be sterilized, and which are mainly done by steaming, okay, by dry heat or by autoclaving at 120 degrees Celsius for 15 minutes to 20 minutes, and this is called complete sterilization. After sterilization, the explant needs to be transferred to the culture medium. That is transfer of explant to the culture medium. So the explants are now transferred to the sterile culture medium, which is either solid or fluid. Okay, the setup is kept under antiseptic conditions or aseptic conditions with optimum conditions for growth. So the transfer or the explants needs to be transferred to the sterile culture medium. After the transfer of explant to the culture medium, it will be followed by the formation of callus. Clear? So in the culture medium, when we will place the explant to the culture medium, the cell of the explant start dividing. It starts multiplying and form an unorganized mass of cell. And this unorganized mass of cell is called callus. Okay? And these cells are totipotent, that means it can form any part, any tissue or any organ of the plant. Okay, totipotent, it has the property to form any cells, any tissue or any organ or part of the plant. That is called totipotent. So callus has got the property of totipotency. After callus formation, there will be organogenesis, the formation of organs, and the formation of plantlets. Okay, so under the influence of various concentration of different hormones, because we are using the growth hormones, all right, in the culture medium, the undifferentiated totipotent callus, that is which is parenchymatous, made up of parenchyma cells. Okay, it will differentiate to form shoots, it will differentiate to form roots, and will give rise to the miniature plants. Clear? And the fate of the growing tissues depends upon the source of selected tissue, that is the explant, conditions for the growth, and the composition of the nutrient medium. So these are the different factors, all right, on which the growth of the tissue depends. So these are the steps, right, or the procedures of plant tissue culture. Now, what is the uses of culture? Callus, which is mainly an or unorganized mass of cells, which has got the property of totipotency, which is mainly composed of parenchyma cells. All right. So callus as well as suspension culture can be used for number one, obtaining cell biomass, which is used for biochemical isolations. It can be used for regeneration of plantlets from the callus, because callus can differentiate from any part of the plant. Production of transgenic plants in which the genes has been altered, okay, which can not be produced by sexual reproduction. Transgenic plants means the plants whose genes has been altered, has been modified, or has been changed. And last one is isolation of protoplast. So for this particular purpose, we can mainly use the culture. Next one, application of tissue culture in crops. Okay, so the usual method of propagation of commercially important orchids by offshoots is a slow process. Okay, and also a time consuming one. It takes a lots of time for development of those plants. Now orchids are raised by the plant tissue culture. In several countries, plants are being commercially raised by the tissue culture. So mainly because over here it will take a minimum amount of time, let's say not long enough of time is in case of conventional technique, but will take a short period of time for the formation of plantlets. So this process can be used to develop those plants which can be used commercially. Okay, so tissue cultures and genetic engineerings are aimed at the selection and the cultivation of new plant which are resistant to diseases, which are resistant to predators, and which can mainly tolerate the environmental conditions also, or harshness like drought, salinity. Okay, and which can also be grown without fertilizers and pesticides. So this is the main aim of tissue culture and genetic engineering, to develop the plants. So therefore we can now say that plant tissue cultures have got the following applications in crop improvement. Number one, in micropropagation, production of disease-free plants, anther culture and formation of androgenic haploids, embryo culture, introduction of new mutations and selection of mutants, introduction of clonal variations, protoplast technology, and somatic cell hybridization. So these are the applications of plant tissue cultures, but we will mainly go through micro propagation. Okay, we'll discuss about micro propagation. We'll discuss about production of disease free plants. Then we will talk about clonal variations, then protoplast technology, and somatic cell hybridization. So in the point number 7, we'll mainly discuss about somatic cell hybridization. So, okay, we'll not go through all of them. Only we'll go through micropropagations, production of disease-free plants. Then we'll talk about somaclonal variations. Then about somatic cell hybridizations, because these are there in your syllabus. Okay. So micropropagations, it is mainly a technique. Okay. It's a process of propagations or vegetative multiplication of plants through tissue culture for agriculture, horticulture, and forestry. So micropropagation is mainly used to multiply the plants that can be used for the agricultures, for the horticulture, or for the forestry. Clear? And therefore the plants which are grown by micropropagations are genetically identical, means they are same to the parents. Okay, the genes will be similar to that of their parents. Now since the plants grown by micropropagations are genetically identical, are genetically similar to their parents, they are also called clones. Clear? So those offspring which are produced by micropropagations, they are genetically identical, so they are known as clones. Okay. So what are the advantages of micropropagations? Number A, it provides rapid multiplications. By this technique, large number of offsprings or planets can be obtained every year. Have you got it? C, the progeny of those plants can be obtained in millions, which multiply with difficult by conventional methods. Clear? So in order to overcome these conventional methods, we are going with the process of micropropagations, where we can develop large number of offsprings. Now cloning can be done throughout the year in a very small space under controlled conditions. Offsprings can be obtained of sterile plants or of rare hybrids of extraordinary characters. So these are the applications of micropropagation. Clear? Okay. Now micropropagation is achieved by the following methods. Number one is multiple shoot reproduction, and number two is somatic embryogenesis. So let us discuss about multiple shoot reproductions. Okay, so in multiple shoot led production, shoot, 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 so shoot tips are being used for the tissue culture and for raising the mini plants. So shoot led production means we are using the shoot for the tissue culture. Now shoot tips in culture medium produces multiple buds, and each buds will grow into an individual shoot. Okay, so by using the hormones which will help in the development of root, the shoot is induced to develop the root. So by using the rooting hormone, which is we are inducing the shoot to develop the root. Okay, so micropropagation of potato, okay, cardamom, raspberry, peach, almond, orchid, banana, okay, is being successfully done on a commercial level. Number two is somatic embryogenesis. The embryo developed from a single somatic cells by the tissue culture are known as somatic embryo or embryoids. That means the embryo which are developed from a single somatic cells by tissue culture, they are known as somatic embryo. Yeah. Okay, so by these two methods, we are able to achieve the process of micropropagation. Number two, the production of disease-free plants. Application, we're talking about application of plant tissue culture. So number two application is the production of disease-free plants. So in plants which are vegetatively developed, okay, which are vegetatively propagated by roots, tuber, bulb, rhizome, because these are the particular offset, let's say, okay, the particular part of the plant which are mainly used for the vegetative propagation of the plant. Clear? So those plants which are propagated or multiplied vegetatively by roots, bulb, tuber, rhizome, in that case the pathogens are transmitted to the offspring. Okay, so along with this particular roots, bulb, tubers, or rhizomes which are used for vegetative propagations, the pathogens will also be transmitted to the offspring or to the progeny. Clear? But if the plants are raised or if they are multiplied by the plant tissue culture from the shoot tips, okay, of such infected plants, they are free of pathogen. That means if the same plants is being propagated by means of plant tissue culture, then the offspring they will be free of pathogens. But if you used to go for vegetative propagations by using the root, bulb, tuber, rhizomes, If the parents are infected, then obviously the offsprings or the progeny will also be infected. Clear? If they are vegetatively propagated. But if the same infected parental plants are propagated or multiplied by the plant tissue culture, then the offsprings will be free of pathogens. Clear? So by this way we can develop the disease-free plants by the help of plant tissue culture. Number third application is introduction of soma clonal variations. It's in the page number 523. Clear? Somaclonal variations are genetic variations. That means there will be changes in the genes observed in those plants that are raised from the callus by tissue culture. So over here, try to break out the word. Variation means changes, differences. Genetic means gene. Okay, so there will be variation in terms of the genes. There will be changes in the genes, mutation in the genes, and mainly these changes are brought by the from the callus by the help of tissue culture. Clear? Now, if these changes, if these variations are economic importance, are of economic use, then let's say what can be the characters of economic importance or economic value, because we are saying that in soma clonal variations there will be genetic variations which can be of beneficial one. What can be those characters which can be beneficial for the plant? It can be that the progeny can develop the tolerance to herbicides, pest, disease, or environmental stresses, or they are overcome the sterility problems. Okay, if the progeny have such characters which are beneficial, then those characters or those plants will be selected and those plants will be multiplied, because those variations are helpful, because it has got the property of disease resistance, it has overcome the let's say problem of male sterility. Okay, it has got the power to resist the environmental stresses. Okay, so if these characters are present in the plant, then those plants will be selected and those plants will be multiplied. Now somaclonal variations are known in most of the crop, let's say wheat, which are resistant to rust and have tolerance to high temperature, rice, resistant to virus and the left hopper, okay, leaf hopper. Then we have got the potato, which have high protein content, and the tomato, which have the increased shelf life. So these are the few examples of the plants with somaclonal variations. Next one, somatic cell hybridization. Okay, so a hybrid produced by the fusion means combining of two genetically different somatic cells. Okay, two genetically different somatic cells of two varieties or two species or genera is called somatic hybrid. So here we are fusing the two genetically different somatic cells of the two varieties or the species or the genera, and the hybrid that is produced by fusing those two genetically different somatic cells is known as the somatic hybrid, and the process of producing somatic hybrid is known as somatic hybridization. So in this technique what we are doing, we are fusing the two genetically different varieties of plants or the species or the genera. Okay, so the hybrid will be produced when we fuse the two genetically different somatic cells of the plant. There are different varieties or different species, then those hybrids are called somatic hybrids, and the process is known as somatic hybridization. Okay, and this particular hybridizations in plant, it is achieved by the fusion of protoplast. So in plants, it is achieved by protoplast fusion. Now, what is protoplast? We are heard about cytoplasm, right? We are heard about the nucleoplasm. But what is protoplast or protoplasms? So protoplast, listen very carefully, protoplast are the naked plant cells whose cell wall is dissolved by the enzyme cellulase and pectinase. One more time. Protoplasts are the naked cells whose cell wall has been digested by the enzyme cellulase, C-E-L-L-U-L-A-S-E. Cellulase is the name of the enzyme that will digest the cellulose present in the cell wall. and the pectinase that will digest the pectin. Okay, so those cells or the naked cells without the cell wall are known as protoplast. So by the fusion of protoplast, we are usually producing the somatic hybrid cells. Okay, for example, we'll go few one. The protoplast from the turnip and the cabbage. Okay, the protoplast from the turnip and the cabbage, and let's say, or between the species of Brassica, Nicotiana, Petunia, and Solanum, between rice and carrot have been produced by protoplast fusion. So here, by the help of the somatic hybridizations, we are trying to develop a hybrid variety of crops. For example, in 1978, you can see over here, tomato and potato protoplast were fused. There are two different varieties. So when the protoplast of these two varieties, tomato and potato, was fused, the resulting hybrid was known as pomato. Okay. Similarly, Burato is an intergenic hybrid between the brinjal and tomato. Okay, so the somatic hybrid of tomato and potato is known as pomato, and the hybrid between brinjal and tomato is known as burato. Okay, so these are the few examples of somatic hybridization. Now, somatic hybrid can be of following three types: symmetric somatic hybrids, asymmetric somatic hybrids, and the cytoplasmic hybrids. Now, what is symmetric somatic hybrids? In this case, there will be complete fusion of both the nuclei and the cytoplasm. So, in symmetric somatic hybrids, there will be complete fusion of both the nuclei and the cytoplasm. In asymmetric somatic hybrids, are the one with the complete fusion of cytoplasm of both the cells, complete fusion of cytoplasm of both the cells, but nuclear genome of one cell remain intact and that of the other cell is represented in fragment in case of asymmetric somatic hybrids. Then in cytoplasmic hybrids, they are formed by the fusion of cell cytoplasm only. The nuclei remain unfused. Clear? So when the cytoplasm and the nuclear plasm will be fused, symmetric. When only the cytoplasm will be fused, nucleus does not fuse, it's called cytoplasmic hybrids. Okay, and when there will be complete fusion of nucleus of both the cells, but the nuclear or genetic material of one cell will be intact and the other cell will be in fragment, then that is called asymmetric somatic hybrids. Now, what are the application of somatic cell hybridizations? First one, it is used in the production of hybrids between the species that cannot be produced by sexual reproduction or sexual hybridizations. It is used in the transfer of genes and the formation of transgenic plants. Transgenic plants are those plants in which the genes has been altered or has been modified by the process of genetic engineering. It is used in the transfer of cytoplasm and the production of allotetraploids. So these are the few applications of somatic cell hybridization. Now, single cell protein. Now, what is single cell protein? Okay, so single cell protein means the alternative source of the proteins for animals and the humans. Okay, so conventional agricultural production of cereals, pulses, vegetables, fruits, okay, will not be able to meet the demand for the increasing human and animal populations. All right, so already more than 25% of human populations is suffering from hunger and malnutrition. The shift from the grain to meat diet is also creating more demand for the cereals because it takes 3 to 10 kg of grains. Listen carefully. It takes 3 to 10 kg of grains to produce 1 kg of meat from animal farming. So we require an alternative sources of nutrition, alternative source of proteins for the animals and the human beings. So these alternative sources of proteins for animals and the human nutrition is called single cell protein. Okay, so single cell protein is the dried microbial cell biomass or total protein which extracted from the pure microbial cell, that is from the microbes, okay, from the pure microbial cell culture, which is grown on agricultural waste and is used as food supplement for the humans or from the animals. The term was coined by Carol L. Wilson. It is mainly used obtained from the microbes. So we have got a few examples of the microbes from which the single cell protein has been produced or has been obtained. From cyanobacteria, spirulina, and chlorella. Bacteria, Methylophilus methylotrophus, yeast Candida utilis, and filamentous fungi, Fusarium graminearum. So these are the few examples of the microbes which are the source of single cell protein, the alternative source of protein for the humans and the animals for the source of nutrition. So this one is very very important. Clear? The sources of single cell proteins along with examples of few microbes. And what are the uses of SCP? SCP means single cell protein. So it is mainly an alternative source of protein, so it can be used as a supplement of human diet. So protein rich supplement for the human diet, it can be used in that form also. It will help in bridging the gap between the demand and supply of proteins for human diet and reduces the pressure on agriculture. Clear? The last one, uses of organic waste and industrial effluents in raising SCP. SCP means single cell proteins will reduce environmental pollution also. So you can see that here we are mainly raising those microbes on the agricultural waste. So we are reducing the environmental pollution also. At the same time, those microbes which are being reared or cultured, it acts as the supplement for the protein diet for the animals and the human beings. Okay, and it will also reduce the pressure on the agriculture where human beings are too much dependent for the sources of protein. So these are the few examples of single cell protein. Clear? So here we have finished the chapter strategies for enhancement in food production. Clear? Now from next class we will begin with the next chapter, that is microbes in human welfare. Clear? So from the next class we will begin with a new chapter, chapter number 10, that is microbes in human welfare. So this much for today. See you in next class. Thank you.